So thank you, Maury, and thank you all for coming. It's a great panel, and it's a panel that comes from all four corners of the globe. So, you know, I think, and that's kind of representative of how this whole iPSC field is growing. And although we're from all four corners of the globe, we're all connected through some form of technology, I think. So I'm going to say a few words, and then I'm going to get the panel to introduce themselves. And Toda-san is going to do a short presentation. But I think that the topic that we're talking about is actually iPSCs getting into the clinic. And I think it's worth reflecting that the short space of time from the kind of initial invention and technology discovery to the point where cells are going into the clinic in formal clinical trials, and the fact that we've got here so quickly. I think the other thing that's truly remarkable about the field is when you look at the origins of it in Japan and the investment in Japan that's been made, not just from companies, but essentially from the whole of the Japanese society, from government right down to the people. That's given an impetus for the development of this industry, not just in Japan. It's special in Japan, but it has really migrated. If you get in a taxi in Tokyo, the taxi driver is going to know what an iPSC is. I think across there. So that's been my experience. I spoke to them the other day, and I'm stealing all their lines. So this is a bluffer's paradise for me. Yeah, and I promise not to use that one. You promised me
you wouldn't say that.
Yeah, that's right. And I think the other thing that's been truly remarkable in the iPS field has been the way that the regulators have responded. First of all, with the changes to get conditional approval in Japan, but also how many other countries have responded with, essentially special treatment for advanced therapies generally. And I don't want to reprise the panel led by my colleague Jackie Barry this morning, but you can see how regulation's been kind of used as a competitive advantage here, really partly stimulated by the move by Japan. So first things first, Toda-san's going to do a 5-minute presentation. He promised me it would be 5 minutes.
Okay.
So
pleasure.
Here we go. A hard taskmaster. Why don't you come and do your presentation?
Okay.
Yeah, sure.
Excuse me.
And then we'll go through each of the panelists.
Okay, thank you very much for giving me a chance to have a small presentation. The purpose of this presentation is just food for our panel discussion, because the title is Key Factors of iPSC Industrialization. So next, this is left side is research, and the right end is market, patient. So in between, there are a lot of factors, very complicated. Of course, here is all things search, also business strategy, regulation, and manufacturing and marketing. Also, in this case, IPS, and this is a very general picture, but I would like to tell the next step. But anyhow, these parameters is like bricks of the bridge. So this part is development, also industry's role and responsibility. Next is, this is just the FUJIFILM strategy overview, but I don't want to make an advertisement of FUJIFILM, but this is just one of the example of industry's examples. So in this case, Fuji's, in this particular environment, self-definition of positioning in development process is very, very important. Whatever players, whatever roles, you have to identify, define by yourself. In Fuji case, key material and engineering technology for cell performance optimization. Cell cannot live alone. We have to organize kind of optimized situation or environment, microenvironment, by engineering technology as an industry. Also, third one is manufacturing. This is also very, very key factors for cost and quality. Last one is business model. Here is, we listed up a little bit, but for instance, key material in Fuji case, iPSC, differentiation cell, and matrix and medium. Also engineering technology, as I already mentioned, microenvironment control. And third one is manufacturing. Fourth one is business model. In this case, participation and collaboration with highly advanced therapeutic partners with our industrial capability. This is kind of Fuji's business model. I want to share with you through the value chains. For instance, Fuji supply as a cell provider, iPSC and differentiated cell. But the center part is using a create candidate. This means seeds. This is normally originated from academia and venture. And also, next to that, we need preclinical and clinical test and manufacturing. As Fuji, we defined itself by ourselves. We have to be self-provider in both iPSC and differentiated cell. And also, right side is CRO and CDO and CMO, development and manufacturing. Next slide shows same scheme, but one example today. Rosu-san also here from Cynata. Together with Cynata, Fuji is just cell provider iPS cell. But also together with iPSCs, we provide not only iPSC, also differentiated cell. Also, we would like to act as a CRO, CDO, and CMO. So case by case, we as an industry would like to be involved. Next one is, of course, cell availability is also very, very key point. I don't want to touch in detail, but we have a collaboration with Bone Marrow Bank in US to supply HLA homotype iPS cell. This is third one. This is existing current pipelines of iPSC derived product for ocular and neurodegenerative part, Parkinson's, and also cardiac disease, heart failure, and cancer. On top of that, also flexible business model and partnership, we provide CDI's iPS cell to Cynata. This is my last slide. So according to this presentation, I would like to share there are a lot of responsibility and role from industry in this particular therapeutic area, because this is really new and revolutionary technology. Thank you, Keith.
Thank you. Perfect timing. Brilliant. Very good. Thank you. And I think one of the things, of course, to make reference to for Toda-san is that not only is he the senior executive vice president and chief technical officer at FUJIFILM, but he's also chairman of FIRM. And chairman of FIRM really promoting the whole industry, and I know that the enthusiasm by which all of the firms have engaged in that is great. And we were very pleased as the Cell Therapy Catapult to sign a memorandum of understanding on Tuesday with firm to promote the industry. So I think that reflects well. So next I'm going to move on to Emile, ask him to tell us about
3 minutes.
BlueRock.
Yes,
3
be
minutes.
happy to.
And of course, formerly of CDI. So those connections
I have a deep affection for everybody on this table up here. And I can testify to that experience. If you in Japan, if you try to give directions to the taxi driver and it's not working, and you can just say, Yamanaka Sensei Labo.
Yeah.
And the taxi driver, there's a good chance, can take you there. That's not true in the U.S.
That's right.
So I'm pleased to introduce you to BlueRock. BlueRock is a platform cell therapy company. We're developing allogeneic, off-the-shelf, iPS-derived cells as medicines. We're targeting previously intractable degenerative diseases like Parkinson's and heart failure with a known mechanism of action, where that mechanism of action is the loss of a specific cell or type of cells, and where we have high confidence that if we could manufacture, we could replace it. So our mission is to replace the cells that were lost in that specific degenerative condition and restore that native biologic function, and by so doing, reverse the degenerative condition, or at least the symptoms of that degenerative condition. It's, when you think about that, a very big vision. We intend to stably engraft a manufactured cell and reverse degenerative disease. That's our goal. Our primary program, our first program, is in Parkinson's disease with the loss of midbrain dopaminergic neurons. We have a secondary program to follow, and that Parkinson's program will initiate in 2018 with our first patients. I hope this time next year to be telling you about our experience with our first patients, if everything goes well and the FDA agrees with us. Our follow-on program is in heart failure. I think we have some very nice investors. We have $225 million Series A financing that came from Versant Ventures and Bayer, the pharmaceutical company. And as investors, they provide a great deal. Both, obviously, we're very happy for their support, but also their strategic guidance. We are a fully independent biotech. We are free to prosecute the opportunity as best as we see fit. So, and we have, some really great nuclei. The founders, Dr. Gordon Keller in Toronto and Dr. Lawrence Studer in New York, arguably two of the most cited scientists in stem cell biology and translational stem cell biology. That's who we are. So,
great.
Thank you.
And I'm sure that Ross is only going to be extremely jealous of the amount of investment that you've had compared to the investment that you've got to get Cynata going in Australia. So Ross, tell us about Cynata and where you are and connections with some of these other guys.
Thanks, Keith. Yeah, the Australian environment represents sheer penury compared to the largesse of investors here in the US, but it means we're very lean and hungry, and that's for sure.
What does that mean?
So I'm actually staying at the YMCA down there. You're all here in luxury. But on a more serious note, as Keith explained, we are an Australian company, even though in a kind of quirk of fate, we're developing technology that originated here in the United States at the University of Wisconsin at Madison. So the connections within Madison are several-fold. Obviously, our process technology originated there, but so too through CDI, now a FUJIFILM company. We acquired certain rights to an iPS cell line, a GMP cell line. Our technology uses iPSCs as a starting material for the manufacture of an allogeneic therapeutic mesenchymal stem cell. We feel that the iPSC route provides a solution to what we believe is a major challenge to the commercial development of MSC-based therapeutics, and that is the reliance upon multiple bone marrow or adipose tissue or other source donors, and also the massive expansion protocols that are required to provide sufficient product. Using a pluripotent starting material obviates that reliance upon multiple donors, and in many ways represents a kind of step change from a reliance upon picks and shovels to manufacture the product through to a much more consistent pharmaceutical business model. So the advent of iPSC technology, in our case, very much opened the door to the final step in the industrialization of an MSC-based therapeutic, and that's the ability to manufacture significant commercial quantities consistently and reproducibly. As you explained, we are an Australian Stock Exchange listed company, and that means that we spend a lot of time on the road, of course, but our activities have, over the past few years since we IPO'd in 2013, have been directed towards establishing that the cells manufactured using the IPS route have all of the right properties for an MSC, for allogeneic therapeutic use, that finally led to us successfully applying to the UK regulatory agency last year to commence a clinical trial, which got underway earlier this year in graft versus host disease. And we are now treating patients in that trial, a total of 16 patients. We aim to complete that in the first half of next year. So from a very exciting perspective, not only for the company, but for the iPSC field, it represents the first clinical trial anywhere in the world using an iPSC allogeneic product. And so obviously there's quite a lot of attention on the outcome of that trial. And interestingly, our experience with the regulatory agencies has been very, very favourable. A few years ago, I think the flat earthists around the world who thought that an iPSC-derived product was going to be a bridge too far has very clearly been shown to be completely incorrect. Yeah. And that our experience, of course, with the UK regulator, which was the fastest route to the clinic, that was very, very good. And likewise, our recent experience with the FDA also in a pre-IND. So we feel that the IPS revolution is now definitely coming of age.
So it's interesting you said, I didn't kind of want to get into the discussion straight away, but it's interesting you said that, well, A, that you're in the clinic in a formal trial, and there's the Flat Earth Society said it wouldn't happen, because I remember those conversations when I first started the Catapult, that it was going to be 10 years away, and it's just not true. It's just got there in a few years.
Yeah, well, we acquired the rights to the GMP line from CDI originally, which is Emile and my connection, in 2014. Yeah. So from that point onwards to getting into the clinic in 2017, and frankly we could have got there in 2016 had the NHS hospital administration's been a little bit more speedy.
Well, I'm trying to fix that.
Yes, I know you are. So that's certainly an outstanding. I'm not blowing smoke in my own pocket, but certainly for a technology which, as you pointed out in the introductory remarks, is very recently evolved. It's certainly a pretty quick path to the clinic and hopefully, eventually to market.
Okay, so we'll get into more of the details of some of those things later, but the final panelist is Hardy Kagimoto from Healios, and Healios, of course, has several programs, including an MSc program with Athersys, but of course you're probably much better known for your work in the IPS space. So tell us about Healios and where you are.
Yeah, so I founded Healios back in 2011. And back in 2011, when I talked to my friends, 10 out of 10 friends told me not to start a company around iPS cell, right? Yeah. So I did talk to myself, and I was convinced, so I started Healios. And I became CEO of the company in 2012. I brought the company public 2015. And we have so far raising $300 million so far. So we are good.
Jealous? Ross?
All of us. Okay. Anyway, so we are doing okay. And then after I brought the company public, I quickly realized iPS is great. Yeah. It's going to come at some point. But realistically, it's going to take time. So also the Japanese regulatory framework has changed drastically. And there was a great opportunity to in-license technology from Athersys team. And it serves the purpose for business-wise and also the patient needs a therapy for acute brain stroke. And we are about to start the phase 3 trial for that technology right now. And then, of course, I started a company around iPSC, especially around the Masayo Takahashi's technology. It's been already 3 years since she has given the very first clinical research for autologous iPSC product for patients who are suffering from wet type AMD. Now she does not need any additional anti-VEGF injections, but the fellow eye, which she didn't inject herself, still requires constant injection.
Right.
So proof of concept is there. But there was an issue with the second patient for the gene mutation. So I think they made a right decision not to move forward with autologous approach, but they shifted to allogeneic clinical research. Now she has given few patients therapy already. I believe it doesn't require any immunosuppressant. So now we're talking about real therapy.
Yeah.
And next step for us is to start official pivotal study for approval for that technology. And we're hoping to get the product approved at some point in a few years' time. And that's where we are. And then also we licensed a technology from Dr. Takebe's lab for technology called organ bud technology, which is a proprietary platform technology to form many type of different organs. That's it's going to take time, but that's a great thing in the future. So that's basically what we do.
Okay, great. So what I want to do now is talk a little bit more about not necessarily just the individual programs that you have in your own companies, but more about the kind of the total pipeline in Japan, and then we'll move on to the rest of the world. So maybe first of all with you, Hardy, and then Toda-san, if we can get your perspective on how the whole industry is going and where the clinical pipeline is in Japan, and then, as I say, we'll move on to the rest of the world.
Okay. So probably I should address why is it really happening in Japan, right? The setting is quite different. We have clinical research system, which is quite unique. Academia can carry out the study by itself. And then those data potentially can be used to justify to start the clinical trial. So these clinical research potentially can be function as phase 1-2 study, followed up by corporate sponsored clinical trials, which can be pivotal study. And that's the structure there. And also there is a conditional approval system. And those who are, if the indication is really tough to enroll the patients, it is possible to obtain conditional approval with limited number of the patients. And the last piece of the puzzle, which sometimes people do not understand here in the United States and Europe, is the government 100% reimburse the product once it's approved through conditional approval. And the difference here in the United States is insurance company pays for it. So no one wants to pay for unproven product. That's going to be the last piece of the puzzle, which accelerates the whole system in Japan.
Right.
And there are a number of clinical research ongoing and also planned to be started around iPS cell technology. So those pipelines can be feeding into corporate pipelines at some point. So.
Okay. So, but the total government investment, of course, in Sera, it has been huge. So you would hope things were going to be coming out, and of course you've had deals like Takeda with Sera to get those. Toda-san, what's your perspective on kind of that total picture and where the next clinical pipeline, where the clinical pipeline's coming, the kind of indications you expect to see moving forward in Japan?
Yeah. As we shared already this conference first days, Japanese regulatory system is triggered kind of regenerative medicine cure therapy, but the so-called conditional approval, time-limited approval system. So that is really opening door for real therapy treatment using cell science, including iPSC. But the next question is, how do we show real performance? Therefore, after conditional approval, the Japanese government and our industry, and also including academia, as a Japanese total society, try to get so-called post-marketing data accumulation. This is really key.
Right.
So then we could identify what disease area is most suitable for instance, iPSC application, and also we could find kind of next agenda, next obstacle to be tackled. So personally, I could tell, especially iPS cell, is not only iPS cell is needed. For instance, other cells, MSC, or other type of alternative. Also, we have to carefully evaluate. But anyhow, first, within 3 or 4 years, this kind of experience accumulation, we could find most proper, in the beginning stage, of course, science is always improving drastically, but in the beginning stage, I think iPS could take some position in incurable disease area.
Right.
So must be very, very careful to identify this area. Otherwise, we have to carefully iPS cell technology make them grow up. So in that sense, a proper area should be very, very carefully watching and searching. This is one. And second part is, as I mentioned already, combination with engineering technology.
Yeah.
Because cell cannot live alone. We have to organize most proper, optimized, three-dimensional microenvironment. This is also kind of parallelly we have to develop.
Yeah, I mean, I was very impressed when I was at your lunch the other day, and I didn't know that so many engineering firms were trying to put together pieces of automation on that. That was quite surprising to me. But of course, you know about all of that in firm. You know, all the big companies are doing that.
That is one of the main purposes. The firm is not only just a friendship organization. We have to bring our own individual strong point of technology. Then kind of total performance could lead this really revolutionary cell into the patient. That is our kind of policy.
Okay, great. So I've got one more piece, a short discussion about Japan before we move rest of the world, and it's a kind of linking piece because, at least from my perspective, the iPS revolution seems to have stimulated Japanese firms to go global more quickly. Do you think that's correct? Because I've never seen the kind of acquisitions that they've been making. I mean, obviously, CDI was acquired, so that seems. Do you think that's part of it?
Yes, sure.
Okay.
Because only single technology or single strong point cannot feasible to reach quickly and at the reasonable cost and the deserved quality. Therefore, we need borderless collaboration is necessary.
So Emile, you've obviously trying to develop a pipeline, but you've had experience before of that kind of globalization. How do you see that kind of global spread going, and how are you going to work your angle from BlueRock now?
I think this panel is a perfect illustration of that. We have two leading Japanese companies, a leading Australian company that's partnered with a Japanese company, and a U.S. company. So I think that speaks to that historic, sort of activity level. And I think that was driven by, the early science, the research, Dr. Yamanaka pioneering it, and then the community in Japan just embracing it and understanding it, the economy, the government. But I'm tremendously excited about outside of Japan, and I think that's good for the world because we need more companies, big companies like the quality of FUJIFILM betting on these things. But I'm tremendously excited about what's happening in the U.S. today too, and I think it's a direct response in a way to how fast Japan recognized this opportunity and started moving on it. Things like the 21st Century Cures Act, and those concepts that are changing the way we think about these medicines, trying to improve the regulatory framework. So I think that's all that, and that might actually have the effect of reversing that flow of some of that money and expertise into the more active environment in Japan. It might actually result in a more balanced flow because today, clearly, there's more flowing in and more activity in Japan than into proportionally. But I think 21st Century Cures might have an effect on that. Our goal is to run the trial. Our first trial will be run in the U.S., be associated with Cornell and Memorial Sloan Kettering, so in New York. We haven't picked the second site for the second trial for heart failure. It could be in Toronto, associated with Dr. Keller and the whole Toronto network. It could be in California. It could be. So
what's the kind of preparative work that you're doing to get the Parkinson's and the heart failure trial kind of position right? Because obviously you're a new company, straight out the box.
Right.
A lot of money, but that doesn't make the problems go away, does it? In fact,
it doesn't.
It concentrates them.
You still have to be very careful. Nobody can afford mistakes. They're very costly. We're fortunate that the Parkinson's program was an ongoing mission for Dr. Studer for most of his life, and we're sort of a late stage partner to his mission to translate the science into a therapy. So he had been funded to do a lot of this work, and he had learned from all the fetal cell transplants and all the trials that had been happening everywhere. They've been happening all over the U.S. and Europe, many different sites. So his trial was very well architected.
Right.
When BlueRock joined,
because
partnered with him.
you've got a model you can back into.
Right. We learned a lot from the fetal cell transplants. We know about patient selection we're better at, the surgical techniques, the immunosuppression protocols, the cell identity. There's basically every aspect of that, and a big element of it being patient selection. Who are the right patients to select? So Dr. Studer has been thinking about that effectively his whole career with Dr. Tabar, and so ours is focused on the movement disorders clinic at Cornell. It's based on all of that, the design. The heart failure trial, we have the luxury of not having to closely define that yet because it's a little bit behind Parkinson's. So.
Right, great. Now, the last but not least on this, and I was privileged. I think it was during the ISCT in London last year, wasn't it? When the announcements came out around your first ever clinical trial with an iPSC, and I said to you on that day, I thought you were getting under-recognised, and I promised to go back and tweet, which is what we did.
It wasn't exactly.
No, it was a bit ruder, that thing, but so we did tweet the bejesus out of it.
Yes.
And tell us about. I was surprised, frankly, that it kind of came from nowhere, and it was in the UK, and we know UK is a good environment for these things, but it was such a surprise for me that out of nowhere this trial started. So tell us about your experience, about how you actually got there, and the kind of what the interactions with MHRA were like to get to that point, because I'm sure everybody here is really interested in that. The very first ever clinical trial, and you did it, Ross. So tell us about it. No pressure.
Right.
Well, it came down to the jurisdiction in which we could get the trial started quickest. We have in Australia, as we were joking before, very impatient investors whose idea of a long-term investment is overnight. And so their view was that, if we didn't get this technology moving and a clinical trial approved very quickly, then it wasn't really what it was cracked up to be. So speed was of the essence, and we'd had some interactions with the FDA on an informal basis, and I had a reasonable understanding of the path to an IND here. Likewise, we had a reasonable understanding of what would be required in Japan, but that sort of took a little turn, as Hardy mentioned before, with the trial change in Dr. Takahashi's lab. And Australia was out because the Australian regulatory environment, although favourable in many ways, for something as new as this was going to be quite a challenge. So of the kind of important jurisdictions worldwide, that turned us to the UK regulator, the MHRA, and we were very pleasantly surprised at the pragmatism that was displayed. And I think, to be fair to the pioneer companies in cell therapy over the last decade or so, their experiences and their interaction with the agencies and their track record have obviously not been specific to iPSCs, but cell therapy more broadly, emboldened, perhaps not the right word, but certainly gave the regulators globally a whole lot more comfort about proceeding to approve clinical trials. So as I said, the MHRA was very pragmatic, a delight to deal with. So, I would certainly encourage those of you in the audience who are considering clinical programs. Obviously, for US companies it makes sense, but the UK, of course, is a A-grade jurisdiction as well, and our experience was very, very good, and it was remarkably straightforward. To some extent, of course, that's a factor of the preclinical package, and as much as possible, we anticipated the areas of sensitivity of the regulator, in particular concerning safety, which I think we'll probably touch on in a minute, and in ensuring that the data package that we submitted covered all of the concerns that were likely to emerge. Now, of course, you can never anticipate until you get in front of the agency what's going to happen, but in our case, it was obviously a successfully compiled package with a comprehensive data set on all of the characterization, particularly CMC of the product, and that clearly gave us a good head start with the MHRA, and that led to the approval in September, and then the trial getting underway earlier this year. So it was a very straightforward process, and one which, of course, has satisfied our investor audience, and the US and other jurisdictions will now follow.
Great. Okay, so maybe we can move on to, I kind of call this next bit barriers, because the things you've got to do to get there. So for an early trial, the barriers are going to be lower than for a later trial. And the regulation is proportionate to the stage and what have you. But one of the things maybe it's worth a discussion around is the kind of donor requirements, cell lines, GMP provenance. I mean, everyone says things are GMP, and you take the lid off, and often they're not. So can you talk about experiences with where you are on that kind of donor, cell line, provenance stuff? And I guess maybe Emile, but can you kick that off?
Sure. FDA regulations on eligibility, capital E, are pretty clear. There are a few places where you could interpret one way or the other. Our perspective is interpret conservatively. And if you do that, I don't think there's a lot of debate on what eligibility means as a donor for an iPS cell. I think that's actually pretty well established. The testing requirements, knowing, the safety requirements in terms of incoming donor selection, they're pretty well established. You can order and have tested all the things that the FDA requires. iPS has a unique requirement in that you have some factors you introduced and a method to introduce them, and depending on the method and the factors and the combination, you might have unique tests. But those are actually pretty straightforward. Plasmid-based or Sendai-based you can test for. So it's actually, in terms of the donor selection and the manufacturing method, these are they can be handled one step at a time.
Right. Right.
I think the big unanswered question that I've experienced in this field is GMP refers to the manufacturing process. The question is, when you ask regulators, when does manufacturing start? And you can ask 10 different consultants and get every possible answer, and you can ask regulators in every jurisdiction and get different answers. And that has deep implications for where you have to actually be compliant and where you shouldn't. So, does it need to be GMP when you draw, or GTP when you draw the blood? Or can you process the blood in that just in a cleanish environment? Can you make a line and then convert it from a research bank to a master cell bank? All those questions are still things that are jurisdiction dependent, and interpretation dependent. And I think today the looser standard of a process that's research grade that's converted to master cell bank is acceptable, and it's produced some beautiful product that's being used around the world. So I think that standard is applicable today. I don't think that will continue forever.
Yeah, I mean,
because GMP is CGMP. It's current good manufacturing practice, and it means that it continues to improve. So I think that at some point the standard will become GMP process, and then GTP plus GMP, the whole thing. And so it behooves us as a community to think about that vector. And I know, as an example, FUJIFILM is thinking about that in the way they're deriving lines.
So as you know, we at the Catapult, we decided that we would derive a line. We started that in 2013 just to try and get ahead of the game. And we said, well, we're going to make it as GMP from the draw onwards. And that makes it tough, and it makes it expensive, and makes it take a long time.
It's expensive.
And, even the GMP we've got GMP plasmids, the whole 9 yards. And so, but it made us a very costly line, which actually you may have seen in the press. We did a license with Universal Cells today, and that's been great, and it's out with other folk. But I'm not sure that £1 million a line is to make hundreds of lines. Do you think we'll make hundreds of lines?
I think we absolutely will. I think we'll make thousands of lines. Do we need them?
Right.
That's a question we still have to address. Our perspective is there's probably solutions where you don't need them. BlueRock's approach is we believe a single line will do it.
Right.
And we can talk about why. And by allogeneic we mean a single donor, a single line.
Right. Right.
So, but that embedded in what you said were so many points that are still up there. Do you need GMP plasmids?
Yeah.
The European regulators will tell you something different from the U.S. regulators So it depends on what standard you're trying to manufacture to.
I've got a freezer full of them now.
Right. There you go. Yeah. So it's very jurisdiction dependent, but I think the whole community is evolving to a higher standard, and that's what it's supposed to do.
What's your view, Toda-san, on this?
Yeah, already Emile mentioned a lot of details, so I could not add many. But one thing is should be feasible in both industry or manufacturer side and also regulatory side. This is one. So in that sense, this is because of this system is our iPSC system is just started. So therefore, normally the regulatory side is they would like to pick up all in details. That is one thing. So should be balanced. But another thing is I'm a little bit optimistic because in this cell manufacturing is scale.
Yeah.
To compare with, for instance, antibody. This scale is so small. So therefore, if we optimize the small system is rather easy, and easy to keep very strict GP system. So in that sense, I'm still optimistic. So as an industry, we have a lot of challenge in technology, but this is new technology. iPSC triggered not only science, but industrial revolution.
Industrialization of science.
Yes.
Yeah,
so in that sense, this is we have to consider.
Hardy, I mean, you've got a program. How do you see those kind of safety barriers?
So,
and, the dealing with the mutation story. What's your take on that?
but the mutation, no one knows at this moment, but we're going to obviously check all the B core mutation for those cell line we're going to use. And from our point, HLA matching does make sense only in Japan.
Right.
My bet is regulatory body is going to come back and say that these products will be a different product because we're going to give thousands of the patients. That's my bet. So we are using cell line from Kyoto University for Japanese trial, but that's only for Japan. And outside of Japan, we're going to most likely to use iPS cell line we have established in a European suite, PIX-regulated one. And then we are genetically modifying the cell with universal cells.
Okay.
So that's a bet.
Okay.
I might be wrong, but that's a bet.
Right. Yes. So, I mean, there's quite a few bets going on this stealth cells at the moment, isn't there? So, how do you think that's going to play out? I mean, an allogeneic edited line, universal sounds very attractive. It's a kind of holy grail. You know, I like to back more than one horse in races usually, and then lose my money twice. But that's my story. So how do you think that's going to play out? Edited versus non-edited, kind of HLA matched versus just non-HLA matched. That's quite important to get that one right, I think. Yeah. Do you want to cover that?
Sure. I would say that there's no single answer because we're facing a whole series of every disease and every cell type is a different set of constraints. So is it a small dose or large, or is it allo or auto? Huge differences in process science problems. So that with that little box of 2 variables, you have 4 completely different process science problems. The same thing, within if you pick a specific disease, like our lead indication is behind the blood-brain barrier. Retinal indications also have some immune privilege. So, matching or stealth is not as important in those indications as it would be in, say, a cardiac indication, right out in front, bathed in the immune system. So it's context dependent. There isn't a single answer. What you're asking is a risk balancing question, and that's our job. Each of us here is to balance risk. So our company doesn't have a single strategy, meaning our lead indication in Parkinson's, there's 30 years of fetal cell data that shows that matching is not required. Doesn't mean that it wouldn't work better or be improved, but we are confident that it's not required. But other indications, you need some strategy, immune suppression, maybe stealth, maybe. So our approach is context dependent on the cell and the disease we're trying to treat. We believe that it will be that and we've also recently announced the deal with Universal Cells. We think that's a beautiful approach. We believe that it's a combination of some editing of the cell as well as bringing the patient closer to there's the donor and the recipient, and you can produce both vectors. You can do both vectors. There's really good science on that. We're, as a company, betting on all those strategies. And next year, I'll tell you all about it.
Okay. But Ross, you're the man in the clinic. You're in the hot seat. What's your view?
Well, to some extent, we're an innocent bystander to this debate about stealth cells because we're developing an MSC, which has that couple of unique characteristics. The first is this essentially immunoprivileged nature of absence of MHC class 2 expression, and also the fact that the cells don't engraft; they don't persist in the recipient, and so the need to eliminate the characteristics of the donor are not so apparent in an MSC-based therapeutic. So I think it's a fascinating debate, however.
You're happy to
But you're willing to watch the race. Yeah.
Quite right. I mean, at the end of the day, all of these approaches are designed to improve the clinical outcome and enable us with much more effective clinical tools that can benefit the patients, and that's what the end game is. So I think that for that reason alone, if it opens up new doors, it's a fantastic outcome, and both of the programs that the Catapult and BlueRock have got, I think will really lead us to a new generation as well.
Okay.
But in the MSC world, it's not so important.
Okay. So Toda-san was talked about the small scale and the automation and kind of wasn't too worried about cost, but cost is going to be an issue.
Yeah.
And
Sure.
you know, MSCs, you know, the whole point of yours is you want to be able to grow a lot of them. So how's the cost considerations playing out in terms of what you're thinking about what you're going to need to do over the next couple of years to kind of chip away at that issue? Do you want to take it, Hardy, to start with?
Yeah, so the first thing, you should have a single cell.
Right.
That's the biggest cost driver for iPS cell. And then scale up. Not, you know, people are saying that they have scaled up iPS cell up to 20 liter, some cases 50 liter, right?
Yeah.
But no one is using it yet.
No.
No one's there yet. But eventually, I think it would be possible to scale up to, you know, 100 liter and so on and so on. And that's basically, that's the bet. And basic technology is out there. Technology has been tested with MSC already, successfully. The question is whether you can use it for iPS. The characteristics of the cells, as we all know, different, right? Unstable. So then we need additional cell detection technology to sort it out. And those are the combinations. But I firmly believe that technologies are coming together. By the time we get our product approved, it will be there in the years to come.
Okay, I'm going to go down the panel. So, Ross?
Yeah, well, clearly, cost reduction is a huge responsibility for the industry. And having said that, of course, we all know that manufacture of cell therapy products is expensive, especially compared to traditional small molecules. I think advances in materials science, robotics, are clearly going to have an impact on cost. You know, as we get better ways, for instance, of culturing MSCs, in my case, in Cynata's case, in 3D systems, microfibers, whatever, will obviously be advantage to the eventual, you know, cost reduction. Robotics, I think, is a fascinating area. You know, one of the elements, of course, of MSC manufacture is the human involvement, and not that we're trying to do people out of jobs, but obviously the less operator involvement you've got, the less risk of contamination and other issues. So robotic technologies can conceivably eliminate much of the cost elements that come from failed batches and other issues. So it's not just the cost of the actual manufacturing cost. If you look at the cost of a failed batch, of course, that can be immense drain, and that's got to be built into the overall cost of the product when it's in the market.
Yeah. Now, Emile, you've got a secret weapon called Bob Dean's.
Yes, I do.
But apart from Bob, you know, what's your view?
I think that, again, there's no single answer here. It's, again, you know, is it allo or auto? Is it small or large dose? And how long is the manufacturing process? The extreme example here would be Dr. Takahashi's pioneering study that was autologous, you know, with a single batch that took months and months in a clean room. You had to layer the overhead of the entire clean room on that one material. It was $1 million for that product. That same process, but if you made 1,000 doses in this flask, in RPE you could make 10,000 in this, would have divided it by 10,000. So there's huge levers here. So it's really a decision about it's a risk management issue again. You know, in our lead program, cost will not be the driver. Parkinson's is 10 million cells, let's say, just as a hypothetical dose. It's not the challenge. You know, you have a total cost budget, and there's the materials and the labor and the overhead and the surgical procedure and the immunosuppression and all those things. All those stack up. I think part of the challenge is that we haven't had a win yet, too, right? We have not had clear efficacy in this room. And when we see that, when Dr. Studer shows that, I think the conversation will be different.
It'll change,
and there's a lot of room for optimism here. There's some great studies, you know, globally going on. There's interesting models, even in the U.S., where we know we have challenges with healthcare, you know, with outcome-based pricing and things. These are very novel ways to think about it. So, you know, if we have a therapy that reverses a degenerative condition, I think it's going to be a fun conversation to have.
Great.
So,
great.
You know, that's what we think.
Toda-san, your view?
Yeah, the cost or price is really difficult problem. So especially, of course, depend on the countries. Japan is more or less national healthcare kind of insurance system. Also, US is different. Also, we have to consider kind of emerging countries. So iPSC therapy is really revolutionary measure to secure the people. So in that sense, cost is really should be feasible. So at least one thing we have to decide right now, don't follow the price setting procedure of the drug.
Right.
Right.
More or less drug once, when they start a business, once price is set, only decreasing, very little. But normal, almost the same price continue. But normal industrial product like motor car, kind of prototype is so expensive.
Yeah.
So in that sense, but as Emile mentioned already, the numbers and price should be balanced. So in that sense, anyway, at least we have to not follow the drug pricing system.
Yeah. So what I hear from the panel is people in the clinic, I hear great confidence. It's just such a vibrant area that I hope to be here next year, asking the same questions to you guys, and you telling me about all the things that you've done. I've got 3 minutes. Is there time for a few questions? Yeah. So maybe I'll take a few questions.
You can, like this, you can see.
I can't really see it. I'm blinded. No questions? Where's?
No? Over here. Where?
I can't see any.
I can't see it. Where?
Mystery.
What is that one?
Mystery.
Okay, so I can't see any questions. That's great. So I'm going to ask a question. I'm going to ask each of you one question now. The same question. If you could change one thing or get one technical fix that would allow you to go faster, what would it be? And I didn't tell them I was going to do this, so I've really put them on the spot. So Hardy,
I think it's a mindset, right? When we talk about these things, I see fear, worry, concern, and they're expecting us to give you some magic answer that iPS is safe. You can invest in that, right? But there's no answer for that, right? Rather than asking those questions or showing your concern, just commit. Do your daily job, move it forward, get your job done. No one knows the future, right? And no one, we have done it many times with different technologies as a human being. Just do your job and see you again in 1 year.
Right.
And then you'll be convinced.
Right.
That's my message.
Okay, great answer. Ross?
Yeah, education. I think the mindsets that surround the early derivation of iPSCs have kind of corrupted the thinking and the views in relation to safety as a consequence of the original integrative approach, which of course is obviously not relevant from a clinical perspective, but has been superseded with the new approaches for iPSC generation. So people understanding the technology more effectively, which clearly the regulators have taken on board. And as I said before, much of that came from the education that arose from our predecessor companies.
Great. Okay. Emile, what's yours?
Those are good answers.
Good answers.
I would just say, to add to that, a clear regulatory framework, just a clear vision for how we can develop these medicines more efficiently and safely and faster, in the U.S. And I think that's coming. So if I could get that, and I think it's coming, that will have a big impact on our development.
Right. And Toda-san?
Yeah, iPS already changed existing paradigm.
Yeah.
So in that sense, how to adapt our kind of social condition to iPS changed paradigm. That's my answer.
Fantastic. So what I didn't hear, all I heard there was confidence. I didn't hear a technical barrier that's getting in the way of this.
That raises a concern, but
because we're blind. Yes.
Well, I told you it's a bluffer's paradise. So with that and 16 seconds left on the clock, give the guys a big hand. I thought that was terrific.